Good morning, students. In our previous class we had done immunity and its kinds, and today I will be telling you the structure of antibodies or structure of immunoglobulins, which are abbreviated as capital I small g. Now each immunoglobulin is made up of 4 polypeptide chains. Out of these 4, 2 are short polypeptides, 2 are short polypeptides which are called the light chains. These 2 are short, called the light chains or also the L chains. And 2 are made up of long polypeptides. The 2 short polypeptide chains are identical. Same way the 2 long polypeptide chains are also identical and they are called the heavy chains. Now the light chain is joined to the heavy chain, and all the 4 polypeptide chains are joined to each other by disulfide bonds. They form a Y-shaped molecule where the upper 2 ends of the Y are the antigen binding sites, and these are identical. These are identical because they are antigen specific and they bind to a specific antigen and form an antigen antibody complex, which is like a lock and key mechanism. Now each of these chains has a constant portion and a variable portion. So this is the constant portion of both the chains, and this is the variable portion of both the chains. This is the variable portion, and it is this variable portion of the 2 polypeptide chains which differ, because of which there are 5 different kinds of immunoglobulins in mammals and even in humans. These 5 immunoglobulins are IgA, IgD, IgE, Ig, and Igm. These are the 5 different kinds of immunoglobulins, and this is because of the variable portion of the 2 polypeptide chains. So this is the basic structure of an immunoglobulin. Now I will read the important points in the structure what I have explained to you. Each antibody is made up of 4 polypeptide chains. There are 2 identical short polypeptides called the light chains or the L chains. There are 2 identical long polypeptides called the heavy chains or the H chains. Each of the light chains, these are the light chains, each of the light chain is bonded to one of the heavy chains. The 4 polypeptide chains are held together by disulfide bonds, and this gives a Y shape to the antibody molecule. The top 2 tips of the Y-shaped molecule, these are the antigen binding sites. They recognize and bind to a specific antigen in a lock and key fashion, forming an antigen antibody complex. Both the arms have the same antigen binding site, and so they recognize the same antigen. Each of the 4 genes has a constant region. As I told you, they have a constant region and a variable region called the C and the V region respectively. Due to the variable regions, there is a huge variety in the shape of antigen binding sites. Hence, different antibodies can react to different antigens. The stem of the antibodies is formed by the C, that is the constant region of the heavy chain. In mammals and in humans, based on this, there are 5 different classes of heavy chains that form 5 different types of antibodies: IgA, IgD, IgE, IgG, and Igm. Now the 5 different kinds of antibodies, one by one we will be dealing about each. IgA, this is the main form of antibody which is found in the external secretions like saliva and tears, etc. in the external secretion, and even in the mother's milk. And the function of this antibody is to attack pathogens before they reach the blood stream, because they are found in the external secretions. Then second antibody or immunoglobulin is IgD. This is present on the surface of the B lymphocytes. On the surface of the B lymphocytes, and they are found in circulation in extremely low quantity, and they serve as receptors for antigens on the B lymphocytes, also help in the activation of the B cells. Then we come to the next kind of antibody body, that is immunoglobulin E. It is present as membrane bound receptor on basophils. This is found on the as membrane bound receptor on the basophils and also in the mast cells in tissues. This is the location of antibody IgE, and this promotes the release of histamines and other agents in allergic reactions. Then we come to IgG. This is the most abundant antibody, and about 75% of the antibodies in blood are of the IgG kind. And this is the only antibody that can cross the placenta. Most abundant and only antibody that has the ability to cross the placenta. So, because of which it provides immunity, works as a passive form of immunity in the fetus. So, and the function of IgG, being the most abundant in blood plasma and circulation, it helps in the stimulation of phagocytes. It attacks the pathogens, that is both viruses and bacteria, and even the bacterial toxins. It provides passive immunity to the fetus because this is the only antibody that can cross the placenta. Then we come to the fifth kind of antibody, IgM, which is present as a receptor on the lymphocyte surface. IgM is present as a receptor on the lymphocyte surface, and this is the first type of antibody to be secreted during primary response, and it causes activation of B cells and promotes the clumping of cells by causing antigen containing particles to stick together. So these are the 5 different kinds of antibodies. We have discussed its location, where they are found, and the important functions performed by each kind of antibody. We have discussed the structure. The diagram is here. I will be putting up a PDF also showing you the structure and the different kinds of antibodies, their location and their functions. Now we will move on to the next topic, that is immunization and vaccination. Now coming to vaccination and immunization. we have discussed about immunity, and we have seen that active immunity comes naturally to a person after there is infection. After a person is infected with a pathogen, the immunity that is generated by the body after infection is called active immunity. Now, sometimes this kind of immunity can be induced in a healthy person by injecting a pathogen in some form, either in a weak form or a killed form, or maybe a toxin produced from the pathogen is injected to generate immunity, to generate active immunity. This kind of process of generating immunity is called vaccination. So, vaccination was discovered by Edward Jenner, and he is also called the father of vaccination. Now, he had observed that the milkmaids and other dairy workers, those people who were working with cows, did not get smallpox. And based on his observations that these dairy workers did not get smallpox, he said that these workers were infected by cowpox at some time, and because of which they are not getting smallpox. The cowpox virus had appeared as a single pustule and had left a scar, but these people were not getting smallpox. So he did a lot of experiments, and in one of his experiments, which was really successful, he injected the live cowpox virus in a small boy named Phillips, and he saw that that boy developed a single pustule, and after 6 months he injected the smallpox in the same boy, and he noticed that this boy did not get smallpox. So based on this, he said that when mild form of a germ has already produced antibodies or has generated immunity in a person, which results in showing no symptoms of a deadly disease like smallpox. So he said that this procedure of injecting a harmless microbe to confer resistance to a harmful, dangerous one is called vaccination, and he is called the father of vaccination. And the word vaccination comes from vaxa, when vaxa in Latin means cow. So this injection or injecting of a germ or a pathogen in some form in a healthy body, in a healthy person, to induce active immunity, to induce the ability of a person to fight infection, to develop resistance against a disease, is vaccination, or we also call it immunization, because immunity is being generated against a disease. The power to defend a disease is created. The substance that is introduced is called vaccine. So a preparation, that is the vaccine, which is introduced in the body to produce an immune response, can be either a killed or a dead germ. It can be a weakened form of a living germ. We also call it an attenuated living microorganism. It can be toxoids, that is toxins that have been rendered harmless but they still have the antigen properties. So the toxoids of these harmful, deadly pathogens can be introduced, or a mild strain of the pathogen can be injected or introduced into a healthy person to generate a immune response. So these are the different kinds of vaccines that are used. Now the vaccine stimulates the immune system. It not only produces antibodies, but it also generates a large number of T or B memory cells. By memory cells, we mean that cells that have been generated in the blood will remain there. And whenever a second time the same microorganism attacks in full virulent form, these memory cells will recognize that germ, that pathogen, and generate a quick response. So the person will not suffer from the disease, will not contract the deadly disease, because memory cells were already there. So this is the purpose of vaccination, or this is a purpose of injecting germs in the body to respond to the virulent germs, or ability to generate immune response in the body. When the vaccinated person is attacked by the same pathogen, the existing T or B cells will recognize the antigen quickly and produce antibodies to kill, to combat, to face the pathogen, and the person does not contract the disease. This type of active immunity is usually long-lived and lasts as long as clones of memory B and memory T cells are present in blood. Some diseases, just a single vaccination is given, and for some diseases, for long-term immunity, a few repeated doses have to be administered to the person. So before I make a table of the different vaccines that are given from birth onwards to develop immunity, and a lot of new vaccines are being prepared. These days, vaccine is being prepared for COVID-19. Experiments are going on so that a person does not suffer from the disease. I will be talking about that table, but let us quickly summarize what I have already told you. So, vaccination and immunization basically means generation of active immunity. By injecting, or we can say by inducing immunity by injecting immunity in the form of, in some form of the pathogen in a healthy person, and this, when I say some form, it can be weakened germ, it can be a killed germ, or it can be toxoids, that is toxins or harmless toxins produced from the germ, or mild form of the germ. This substance that is introduced is called vaccine, and vaccine comes from the word vaxa, which means cow, because this was discovered by Edward Jenner, and his observations as I told you, were based on milkmaids, dairy workers, those that were working with cows, got the cowpox infection sometime in their life, and they did not contract the deadly disease smallpox, because the cowpox infection had already provided them immunity. So introduction of a mild form of the germ conferred resistance against a deadly disease. So this mild form of germ confers resistance against a deadly germ or a deadly pathogen in immunity, or by the process of vaccination, or that is also called immunization. So now I will make a table here in which I will mention the names of vaccines and the age at which it is given and against the particular disease. So this table is important. First, we will write the name of the vaccine, then the disease, the age at which, and what is the safety of that vaccine. We are writing about important vaccines. First, DPT and HIV. This D stands for diphtheria, P is pertussis, which is also called whooping cough, and tetanus. HIV is Haemophilus influenzae of the B type. This vaccine is given to all babies. We are talking of the age group in which this vaccine is given, all babies of 2, 3, 4 months of age. And the safety of this vaccine is 90 to 99%. Then the second vaccine is Hepatitis B. This is against the disease hepatitis, again given to all babies whose mothers have suffered from the disease. All babies whose mothers or close family members have been infected. Now, the safety of this vaccine is not yet known to us. Then the third vaccine is Polio Vaccine, which is also called the SARS vaccine. And this is against the disease polio, again given to all babies of 2, 3 or 4 months of age, and the safety of this vaccine is 100%. Then fourth vaccine is BCG, Bacillus of Calmette and Guerin. It is against the disease tuberculosis. In short, we call it TB. And this is given to children between 10 to 14 years of age, and safety of this vaccine is about 70%. So this is a table that talks about important vaccines. Now all this that we have discussed till now is about active immunity. Now passive immunity is when pre-prepared antibodies. Now when we inject what is active immunity, I have already explained it to you, but since I am talking of passive immunity, active immunity is immunity which is generated within the body. The antibodies against the antigens are prepared in the body. Passive is when ready-made antibodies are put into the system to fight against a disease. So when a person is infected with some deadly microorganism, you cannot wait for that long for the body to generate immunity. It can be dangerous, it can be fatal for the person suffering from a deadly infection. So in that case, to generate a quick response, pre-prepared antibodies are injected from outside, which have been developed in some other animal, and like in horse or in rabbit, the vaccine was given and antibodies were prepared in that animal, and now the serum containing these antibodies are injected into a person suffering from that disease for a quick immune response. This is passive immunity. And even for snake bite, against snake poison, against snake venom, there are injections which have pre-formed antibodies to give a quick immune response. So this kind of immunity, this kind of immunization is called passive immunization. So this is enough for today, and I hope your preparations are going on in full swing for your half-yearly exams. Good luck to all of you. Take care.